Hello students, welcome to another lecture on Understanding Biology chapter 25 part 2. We are still learning about fungi. In this lecture we are going to learn about fungal taxonomy and characteristics of main phylogenetic groups of fungi. At the end of this lecture you should be able to list the main phyla of fungi, describe the characteristics of Microsporidia, describe the characteristics of chytrid, describe the defining characteristics of Blastidocallidomycetes, describe defining characteristic of Neocalimestigomycetes, describe defining characteristics of Zygomycetes and Glomeromycetes, and describe general characteristics of Basidiomycetes and Ascomycetes. So these are all seven different phyla of fungi phylogeny. So we are actually looking into each of these phyla and being able to describe their characteristics. So let's first look into fungal taxonomy. Fungal phylogeny and classification change often when new information become available. So a complete and accurate phylogeny for fungi therefore is yet to be determined. Fungi are descended from nuclearid protist. So we did learn about protist before. So these nuclearid protists are actually a group of amoeba that have pseudopodia. You learn about amoeba with pseudopodia before. So we believe that the fungi actually descended from these protists. So currently, based on characteristic of meiosis, 4 fungal phyla are identified. So those are Chytridomycota, Zygomycota, Ascomycota, and Basidiomycota. Later, molecular analysis showed that these chytrids and zygomycetes are not monophyletic. This actually led to chytrids being subdivided into 3 more phyla, monophyletic phyla, and now we have Chytridomycota, Blastocladiomycota, and Neocalimestigomycota. In 2007, microbiologists agreed upon 7 monophyletic phyla, including all that is shown here. And now the Microsporidia are sister to all other fungi, but there is a disagreement as to whether they are true fungi. So we are actually going to look into each of these different phyla of the fungal phylogenetic tree today. So let's look into Microsporidia. Microsporidia are small obligate intracellular animal parasites. They are long thought to be protist. They lack mitochondria, but their genome actually contains genes related to mitochondrial function. This actually led to the hypothesis that the ancestors of Microsporidia had mitochondria. Microsporidia contain reduced mitochondrion-like organelles. These actually, together with other evidence, place Microsporidia from protists to fungi. So that is how Microsporidia ended up in the phylogeny of fungi. So a little bit further information about Microsporidians. These Microsporidia, members of Microsporidia, can be parasites in many groups of animals, including mammals, crustaceans, fish, and bird. Microsporidia infection usually occurs via GI tract, gastrointestinal tract, and sometimes when animals ingest these spores, they absorb water, swell, and then rupture in the GI tract. This rupture ejects the tightly coiled polar tube, which can penetrate the epithelial cell. In human, Microsporidia rarely cause disease in healthy individuals, but they commonly can cause disease in immunocompromised individuals. So that then they can actually infect the intestinal and neuronal cells, leading to diarrhea and neurodegenerative disease. In this picture here, you can see that tightly coiled polar tube and their spore. Once the spore absorb water and rupture, this polar tube will release, and this tube is actually can penetrate the cells. So now we are moving into the next phyla. So like I mentioned earlier, until recently, members of this Chytridomycota, Blastidomycota, and Neocalymastidomycota were thought to be one single phylum. This was because they have their common characteristic of flagellated zoospores. Flagella have been lost in all the other fungal lineages except for Microsporidia. Therefore, the paraphyletic phylum of Chytridomycota was separated into 3 monophyletic phyla. So, members of the phyla Chytridomycota have aquatic ecology. Their production of motile zoospore is a distinguishing characteristic of this phyla. They have close relationships to basal lines of fungi such as ancestral fungi. As we discussed earlier, Batrachotochytrium is involved with amphibian die-off. We talked about it earlier, while other chytrids have been identified as plant pathogens. So this group of fungi is very important in that sense. Now let's move into another phyla. This is also closely related to chytrids, the Blastocladiomycetes. Blastocladiomycetes are found in both aquatic and terrestrial environments. Members of this phyla are parasites of plants and invertebrates, algae, and other fungi. Most distinguished feature of Blastocladiomycetes is their alteration of multicellular haploid and diploid life cycles. They alternate with a diploid spore-producing sporophyte with a haploid gamete-producing gametophyte. Both generations are multicellular, while sporophyte produces spores by meiosis and gametophyte produce gametes by mitosis. Now let's look into Neocalymastigomycetes. Members of this phylum lives within the rumens of mammalian herbivores. We did talk about them earlier when we talked about the relationship of fungi. They enzymatically digest cellulose and lignin. Sheep, cows, kangaroos, and elephants actually depend on these fungi to obtain sufficient nutrients. They lack true mitochondria and use hydrogenosomes to produce ATP. Genes of Neocalymastigomycota actually obtained from horizontal gene transfer from bacteria actually. These fungi actually can survive on cellulose alone. So these genes encoding digestive enzymes such as cellulase were obtained from bacteria through horizontal gene transfer. So because they have the ability to digest lignin and cellulose in anaerobic condition in high temperature, they actually have potential in biotechnology for biofuel production using these grass with high cellulose content, and it is one of the venues that a lot of people are now doing research on. Now let's look into phylum Zygomycota. So earlier I mentioned that Zygomycota are polyphyletic. They are not monophyletic. This phylum include only about 1,050 named species. They are incredibly diverse. Members of this phyla are common bread mold species found in decaying organic matter such as strawberries, and few members are pathogens on plants and animals, including immunocompromised humans. They have predominantly choanocytic hyphae. If you remember what are choanocytic hyphae, hyphae are continuous and not separated by a septum. When conditions are unfavorable, chemotropic hyphae of opposite mating types attract one another and have sexual reproduction. So let's look into one of the example of a member of Zygomycota, which is Rhizopus. Their sexual reproduction is represented in this figure here. So alteration of hyphae of different mating types lead to gametangia formation. You can see in this picture gametangia with multiple haploid nuclei fuse, and haploid nuclei fuse in the process called karyogamy. So this is a new word for you. So when these haploid nuclei fuse, this process is called karyogamy. These fused gametangia containing diploid nuclei now develop into a zoosporangium, where zoospores are developing. These spores germinate to produce haploid hyphae growing to a new mycelium. So this is the life cycle of Rhizopus, one of the popular members of Zygomycota. Now we are moving into next phyla, which is Glameromycota. This is a small phylum with only about 150 known species. But glameromycetes are important for two reasons. One, they are obligate symbionts with plant roots where they form arbicicular mycorrhizae. And second one, because of their symbiosis with plant roots, glameromycetes were likely have helped plants to colonize land. There are no sexual reproduction in this phyla. So we did talk about the mycorrhizae earlier, like how do they produce arbicicular mycorrhiza and what kind of plants do they produce this kind of relationship. So the fungi that produce arbicicular mycorrhizae belong to this phylum called Glameromycetes. So moving into our next phylum, we are actually going to look at Basidiomycota. So Basidiomycota, you might have heard about it already. So there are a large group of fungi with over 30,000 species, and they include common edible forms of mushrooms. They also include many important pathogens, as well as majority of life cycle spent as monokaryotic hyphae. We did talk about monokaryotic hyphae, dikaryotic hyphae, so refresh your memories. So majority of these fungi actually live their life as a monokaryotic hyphae. When different mating types of monokaryotic hyphae fuse to form a dikaryotic or secondary mycelium, this type of mycelium is called heterokaryotic, with two nuclei representing two mating types between septa. This dikaryotic stage in the life cycle is common characteristic of many fungi, and it is found in both Ascomycetes and Basidiomycetes. So let's look into their sexual reproduction cycle. So due to environmental signal, a secondary mycelium is triggered to enter to the sexual reproductive cycle. This reproductive cycle is characterized by formation of dense above ground fruiting bodies that you have seen. Late in the fruiting body development, some hyphae develop into a reproductive club-shaped structures called basidia. Karyogamy actually occurs within the basidia, giving rise to a diploid cells which undergo meiosis to produce 4 haploid spores called basidiospores. These basidiospores can travel through wind and land on a moist environment and start a new fungal hyphae. Last phylum of fungal phylogeny is Ascomycota. So Ascomycota actually contain about 75% of the known fungi, and it is very diverse. They contain economically important fungi such as bread yeast, common molds, morels, cap fungi, and cup fungi and truffles. This phylum also hosts many plant pathogens. One of the most famous ascomycete could be Penicillium, which was accidentally discovered by Alexander Fleming. Asexual reproduction is common in ascomycetes. They produce asexual spores called conidia in their conidiospores. And we are going to look at the sexual reproductive cycle in ascomycetes. The ascomycete sexual reproduction is initiated when two compatible hypae make contact. One hypae produces antheridium while the other produces ascagonium. Their fusion results in mixing the cytoplasm from each hypae. This will produce a protein body called ascocarp. Certain hypae in ascocarp develop into a structure called asci, and karyogamy happens, which result production of a diploid nucleus. This diploid nucleus undergo meiosis to produce two haploid cells, each of which then undergo a single mitosis to produce 8 haploid ascospores. So here is a picture of cup fungi and morel fungi, and then they are also showing the sexual reproduction, the production of the ascospores through their life cycle. These spores actually can go and start a new fungi. Mycelium in a good, when they find a good moist environment. So some ascomycetes are single-celled. So the most common one that we all know is yeast. Yeast are actually ascomycetes with which they reproduce asexually by cell fusion or budding. Sometimes two yeast cells of opposite mating types can fuse to form a dikaryon. This cell may then function as an ascus. And we all know that yeast is economically important as they have the ability to ferment carbohydrate, breaking down glucose to produce ethanol and carbon dioxide. This ability is used to make bread, beer, and wine, and many different strains of yeast are selected and bred for these different kinds of process. And we all know about Saccharomyces cerevisiae, which is also used as a model organism. Yeast is long-standing model system for genetic research. First of the eukaryotes to be genetically engineered extensively, and Saccharomyces cerevisiae is the first eukaryote to have their genome sequenced, and other fungi are being sequenced and studied, which may affect our understanding of their evolution. So I have a table 25.1, that actually shows the groups that we just talked about, typical examples and key characteristics and approximate numbers of living species. So I thought this is actually a good table for summary of what we learned today. So in summary, we learned that fungi are descendant from nuclearid protist, and they contain 7 monophyletic phyla in its phylogenetic tree. The first one we did talk about, Microsporidia. They lack mitochondria but contain mitochondria-related genes, and they are actually the sister group for all the other fungi. 3 close-related phyla, Chytridomycota, Blastocladiomycota, and Neocalymastigomycota, have flagellated zoospores as a common feature. Zygomycetes are named for the production of diploid zygotes nuclei via karyogamy. And we did talk about glomeromycetes, which produce actually obligate symbiotic relationship with plants, may have made it possible for terrestrial plants to evolve. And lastly, we did talk about both basidiomycetes and ascomycetes. They produce protein bodies through their sexual reproduction. I hope you enjoy this lecture. Until we meet with another lecture, have a good day. Thank you.